Good morning, everyone. So I'm Will Tanadet, Pipatpolkai, and it is my great pleasure to give a talk today. It's my third time at the Sophian meeting, and it has been my greatest opportunity to come to Japan every year and to discuss some of my scientific research work as a computational scientist. Now, in my talk, I'll be mainly talking about the role of the PIP2 lipids and how they open certain families, not just one, families of ion channels. And we'll be looking at using MD as a tool to look at that. And if you cannot remember my surname and how to spell it, it has PIP2 in it, so that might be able to be useful when you try to search my name, such as PIP2 and then Will or something. Okay, so let's think about PIP2. And the story about PIP2, I think, begin around 1996. Right? So PIP2, just to remind everyone, is an anionic lipid found about 1 or 2% in the membrane composition, and they regulate ion channel. The first paper on how PIP2 regulate ion channel, which was ATP-sensitive potassium channel, was done by Don Higelman and Rebecca Ball, where you can see here that having a PIP2 increased the sodium-calcium exchange. And by abolishing PIP2 using phospholipase C, you decrease the sodium-calcium exchange. And then followed by several work on the KATP channel as well, done by Xiaoling Xing and Tom Balkowitz, showing that PIP2 increased the open probability of the channel and therefore increase the firing of the channel. So what we get here is that PIP2 has been shown to activate KATP channels and we know that for a long time using electrophysiology. Now, taking from that time back to the present day, we have many more work, and it's not just KATP channels now, but we have used PIP2 to study the activation of other family of a channel by using the DiPCA PIP2. Also, some of people have been getting lots of structures of ion channels with PIP2 bound, those such as the KR2.2, which was done by Rod McKinnon's lab, and KCNQ1, which was done by Ji Sun and many others. And they show that PIP2 doesn't just stick to the channel, but they also introduce key conformational change to the channel. So the channel, it's not just having PIP2 stuck there, forming a normal lipid, but it also involved in the conformational change and the gating of ion channels. In some channel, PIP2 also involved in the inhibitory role. Now, of course, the issue with PIP2, and I think Ben will have a poster on it at some point as well, that not all the structure that has been solved comes with PIP2. And also, if you're a structural biologist, we know that by having structure, not all of the structures are solved in the open state. And here I'm showing lots of TMEM16A structure, which was done by several labs. Christina Paulino's lab, and also Raymond Dutzler and Andy Lam and many other people were trying to solve the open state of TMEM16A, and they couldn't get the PIP2 density to be formed. So there's so many work on it, but none of them are PIP2 bound or open. So, and if you use AlphaFold, you will probably get the structure, but not with the PIP2 bound unless you play some, very fancy AI tricks that is coming in this new era. So what can we do to it to get the structure with PIP2 bound and actually open so that we can work on it? Now, I'm bringing you to a tool called molecular dynamics, and it's pretty simple. The idea is that you take the structure deposited on the protein data bank or the PDB. What you then do is that you then embedded your protein structure that has been solved in the phospholipid bilayer, solvate them in water, and then add the parameters to it. So you can add the bond length, bond angle, dihedral, the electrostatic, and the van der Waals. And then you integrate all, and because you now put in the parameters as the charge, the bond length, you can get the force, and with F equal ma, you can end up with a molecular movie like this, which is the trajectories. So you can think of it when you solve protein structures, it's like a frozen tuna out of the sea, and then what you then do is that you put the frozen tuna back into the environment with where home, which is the lipids and water and salt and everything, allowing the dynamical motion, the movement of the ion channels to be studied. Now, So the issue with MD is that when you do MD, you get the timescale limit. So when you run traditional MD, you probably get capped at about 1 or a few microseconds. Now, the most interesting ion channels event that we want to study is within milliseconds event. The gating, the dynamical motion, the conformational change of the protein is in the milliseconds event. But our tools, the integration of it, you can do so many of them, it is limited in the microsecond. So how can we get out of that bottleneck? Because if I were to keep running the long 1 microsecond MD, it would take me probably 10 years, and no grant would let me spend 10 years using one long simulation. It wouldn't work that way. So you need to break the time scale somehow to get to this. And one of the technique is the coarse grain MD, where instead of simulating every atoms in the system, you combine the non-hydrogen atoms, 3 of them together, to make one particle, allowing less atoms in the system to be calculated and also reduce the number of water, and therefore you have a smaller atoms to calculate and using a bigger time step as well. The other technique that I'm mainly going to be talking about today is enhanced and adaptive sampling. So you can favor a transition from one state to the other state. And you have a structure get stuck at the bottom of the well. There are several strategies you can do it. One is that you can seed the structure. So start running simulation from different points along the transition. So let's say I'm moving my arm from down to up like this. So instead of waiting for my arm to go from down all the way slowly moving to up, I can force it, putting some lever or something like that to push my arm to go from one state to the other state. The other way is, and by doing so, I can get, I can calculate the force required for me to transit. But that's not very physiological, because who moved your arm like this, right? You can move your arm like this, or you can move your arm like this, and you can get to the same place. The other way is that you can take several snapshots along the way. So you can go like this, 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 and let your arm naturally move like that. You can imagine? Yeah, maybe. And then you try to stitch them all together by looking at the transition between your arm movement. And that's what we call adaptive seeding along the path. Okay, so my ion channel, what I'm interested in for this project is an ion channel called TMEM16A, which have this unique anoctamin fold. This is calcium activated chloride channel, which are found in vascular tissues. These are activated by calcium and PIP2 lipids. And it's very important because it is on this cell called the pericyte. And if you have ischemic stroke, you can have calcium influx inside the cell, leading to chloride ion coming in, and this leads to pericyte contraction. This is quite important because if you can design a pore blocker to this, you can see here where you have before the stroke, you have a blood vessel as normal. After stroke, you get a contraction of this blood vessels coming to your brain. Now, if you have a pore blocker such as ani9, what you can see here that after a stroke, the blood vessel is not contract anymore. So you can target this ion channel and therefore use it to treat stroke. And that's what we are interested in. Now, of course, I'm interested in looking at how you can go from one state to the other state. And the key player that I'm going to show you is PIP2. But how can you find a PIP2 binding site on the ion channel if we do not have a structure of this TMEM16A with PIP2 bound? The traditional approach to this problem is to dot PIP2 around the box. So 10% of PIP2 and 90% of PC, which is a general mammalian lipid. And you run several coarse grain simulations, allowing lipids to equilibrate to the binding site. And you can see here that this is done in coarse grain because my carbon doesn't look like a carbon anymore. It's just a blob. So instead of 16 carbon, I end up with 1, 2, 3, 4, circles forming a lipid tail. And this was done by Martini too. And you get this PIP2 binding site located in the interface of TM3 to TM4. And if you convert from the coarse grain representation to an all atom configuration, so i.e. having all the carbon being represented, all the phosphate being represented, and run equilibrium MD, you get these residues in the binding site. And by comparing that to the previous experimental data, this one done by the group in the States, you will see that the mutation has an effect on the PIP2 binding site, reducing the T-half. To get to the open state, instead of just running simulation and finding the PIP2 binding site, we need to favor the transition from closed to open. And this is what we call, and this is a strategy called fluctuation amplification of specific traits, or FAST. So what is FAST? FAST is a system where you run several replica of MD simulations, so 10 replicates of it. And each replicate would have different initial velocity. What does that mean is that they will take a different path. If you have watched anime called Steingate before, or have you know the butterfly effects before, you know that little things that happen in your life can have a long run effect in your life as well. And here, in the slight difference in the initial velocity, who leads to different outcomes of the structure. What you then do is that you take different snapshots from these outcomes, and then you rank, you cluster the structure. So you cluster the structure based on the RMSD, so how different they are from each other. From clustering, what you then do is rank them. So what do you rank the structure on? You can rank them on the two key parameters. A is how open they are. We want the open state, so we want to move towards the open state. So you favor how far the pore is open, right? So that when you reseed and continue the run, you can get to the open channel. The other favorability, other factor that we choose is the rarity of the state. Because if you get the open state, but they are very stable state, you get stuck at the local minima. You never get out of it. So you need to balance between exploration and the rarity of the state. And that allows you to move towards the more open state. And by doing 15 generations, so 15 cycles of this with 10 replica towards the open state and 10 generation favor the closed state, you get the full free picture of a free energy landscape during conformational transition. Now, why is this helpful? So if I have three systems here, so calcium-bound TMEM16A with PIP2, calcium-bound TMEM16A without PIP2, and no calcium and no PIP2. And we look at the key distance at the outer gate, so V543 and I640. And by combining all the trajectories together, taking these different snapshots along the way, favoring along the way, you can stitch all the trajectories together and plot these free energy landscape. You can see here there's a yellow star correspond to the closed state, the green star correspond to the intermediate state, and the purple star correspond to the open state. Now, you can see here that if you have no calcium, no PIP2, there is no, and looking at the distance, there is no open state being sampled at all on the free energy landscape. If we take with calcium, but no PIP2, you can see where the purple star, the open state is, which is about here. You can see that it's not at the minima. But if you have calcium and PIP2, your open state here is located, there is a minima that you can extract from the structure. If you take that structure and you apply electric field at different values from 0 to 100 to 200, 300, 400, 500, and then you apply electric field and count the number of ion moving across the membrane over 500 nanoseconds, what you can see is that the number of ion permeation correspond very well, very closely resemble to the experimental conductance done in 2008. What that suggests to you is that if you don't have PIP2 in the structure, it is very difficult for you to get access to the stable open state for you to do the conductance study and to work on it, which is the big problem when you try to study ion channel simulation and they try to collapse to close. But it's not just about obtaining the open state structure or seeing what they look like. It is also about looking at what are the key dynamical motion within the structure that takes place. So instead of looking at the distance now, I'm interested in this hinge angle on the TM6 of the protein. So looking at the angle between these three residues. And what you can see very clearly is that if you have no calcium, no PIP2, you do not sample anything above 160 degrees, whereas every other in the presence of calcium, all of these are being sampled. What that suggests is that calcium is important in the kinking of the TM6. And this TM6 kinking is known as the steric gate, or the lower gate of this ion channel. So there are two gates, one at the outer mouth, which is at the top that I've just shown you, and one at the lower mouth in the lower region. And this telling us that the opening of the outer gate is coupled to the TM6 helical motion down in the lower gate of the channel. Now, if I show you that this technique works, it would be great. But if it worked for only one channel, it's not so great, right? So we need to show you that it won't work in more just one system. And this is the work done by my student in the lab called Shiral, and we are working on KCA 3.1. The fun story about KCA 3.1 is that, so when I was a student, let me tell you a quick anecdote for about 1 minute, is that I told myself that I will work on potassium channel, and I've been working on the K2P channel family with Stephen Tucker, KAR channel, and also the KV channel with Lucy. And I said, I will never work on KCA channel because they're too big, they're too complicated, and it would be so difficult to do any simulations on it. And then I came to this Sophian meeting, and people kept telling me to collaborate with George Chandy repeatedly. And I was like, please go and work with George Chandy. He has the Q patch in Singapore. And then I emailed him, and then he said, it would be nice to collaborate. What channel do you work on? The KCA channel. So, everything can be stemmed from this meeting, and then you get to work with the endless possibility here. Anyway, this KCA channel is activated by voltage, calcium, and PIP2, and the mutation on this causing ADC is called hereditary xerocytosis. And there is no structure with PIP2 bound on KCA 3.1. For 3.2, there is some model that people have made before. So the main problem with potassium channel that we had when we do simulation work is that they tend to collapse if they are not fully stably open. So you have a structure with an open state, you run a very short simulation, it will collapse to place. And then you can't really do ion permeation or any other study with it, and it becomes a problem. So how can we get the open state to be stabilized? Well, again, we need to identify PIP2 binding site. And this time, instead of using coarse grain simulation, we took the coordinate of PIP2 from KCA 3.2, and we run short MD, and we run 500, and we run MD simulations on this. And here we obtain the key binding site located here, and it's the same residue as the KCA 3.1, and they bind very stably, as you can see here with the low RMSD at about 3 angstrom. So this suggests that the PIP2 binding site is highly conserved amongst all the KCA channel. But it's not just that. It also helps stabilize the open state. So by doing the same thing of adaptive sampling and plotting the free energy landscape here, the open state is located just here. The closed state is denoted on as a red star. It's usually located on the maxima of the channel. And you can see here that the closed state is also here. The open state for the structure with PIP2 is in the bottom left here. And you can see that it corresponds to the most open conformation. So dark blue suggests the most open, gray is the least open. And if you can see here without PIP2, you cannot get the structure with the dark blue patches on the free energy landscape, suggesting that PIP2 help you to gain access to this conformation of the channel. Now, what is also very neat is that instead of just having a structure like that, what we can see that by taking the snapshot from the open state of the KCA 3.1, not that it doesn't collapse, we can show here that not just it doesn't collapse, and it also allows the potassium to permeate across the channel and knock the other potassium in a selectivity filter, suggesting that that specific conformation is really the open state. Of course, this is very preliminary. More work on this will be shown in the poster, and we can come and talk about how you can open your favorite channel, and we can work on this together. So to conclude, PIP2 can be used to facilitate the access to the open state of an ion channel in MD simulation, and it doesn't just work on the TMEM16A family, it also works on the KCA family. We can use adaptive sampling to break the traditional timescale, stitching all the simulations together. And of course, by getting to the open state, not just that we can look at the static snapshots, but we can now look at how activators bind and opens the channel. So not just the affinity, but we can calculate the efficacy of how the activator may activate the channel. Lastly, I would like to thank my team, Sharal, who has been working very hard, George, Miao Shang, and Ong Xiao Theng, who work on KCA 3.1 and gave me a lot of insight, LQI Fellowship, NTU-HPC, and Sofian for letting me give you the talk. Any collaboration, contact me via email. Thank you very much.
Thank you. So now this presentation is open for discussion. Any question or comment, please?
Thanks. That was a very interesting talk. So I'm just wondering about, so I'm not in the potassium channel field, but I kind of follow it because of all the important work that's been done there. But if you look at prokaryotic potassium channels, the first ones that were really studied structurally, they don't need PIP2 to be gated.
No.
What happened during evolution when you get the diversification of potassium channels so that now they had this requirement for PIP2, or at least some of them had this requirement for PIP2. So what do you think happened? What was the, I guess it's asking, what does PIP2 do? Because you say it collapses the pore if it's not there. So what happened during evolution that made that happen? And
yeah, that is very interesting because, so I just show you Team M16A work, right? And one of the structure, the very closely related protein called Team M16B, right, look exactly the same. When PIP2 bind to it, it closes the channel. So it does the completely opposite thing. Actually, that's what I wrote in my next round, that I want to understand how and why evolution works and why PIP2 can have two different roles. But the question that you asked is, how does it work in prokaryotic, right? And there was, I think, some work done by several people that in prokaryotic, there is this, there's a lot of, not chemolipin, what's it called? Cardiolipin, right? Which is the four-tail lipid. And in those channels that I've heard, cardiolipin may also regulate the channel, and they're also charged in there. So there may be some toggle switch there that evolve instead of having a more charge dependent. So PIP2 is very charge dependent, very anionic. And for the cardiolipin, it's very non-charge dependent. It's more van der Waals space dependent. So I think there was some pocket evolutions around there. So I think it still needs to be regulated by lipids, but also the different kind of lipid pockets that is.
Yeah, I don't want to hold your time, but it's just because, GPCR signaling, in some kind of GPCR signaling, generates PIP2, right?
Yes.
So I'm wondering if the occurrence, the diversification of that signaling coincided with the requirement for some channels to have PIP2 regulation. So I mean, a kind of co-evolutionary thing.
Yeah, because for case in Q1, it comes with the GPCR signaling as well, and those are PIP2 dependent. So in some cells, and that was the work done by Ji-sun as well, that you can see that some of the channel that has lower affinity to PIP2, so they're more dependent on PIP2 tuning, those are tunable by PIP2 concentration. But there are some which are almost always going to be bound by PIP2, and those are not prone to be regulated by GPCR. So yes, there are some correlation and regulation where GPCR is regulated by PIP2, and then PIP2 regulates the channel, and then you have the channel which are fully regulated by PIP2 itself. Yeah.
Okay, thanks.
Any others? Oh. First, then.
Thanks. Very nice work. I just wondered that you got this open structure by running the simulations, but I mean, I'd like to see how the open structure is different from the closed structure. Do you have that information in your slides?
How does it different from
Team M16 with calcium and PIP2, you got this open state, but there's no direct comparison between the closed.
I didn't show the alignment.
So
no, I'm sorry, I didn't. Sorry, yeah, I didn't have the alignment of this, but I have the KCA if that helps you. This one, you can see, I didn't take the whole thing, but I can show you just the gate of the pore, where if you look at the red state, which is the closed state, is here. And if you look at the region here, the outer mouth of the pore, they are the conductive state. So it's more open just there. For TMM16A, there are also some rearrangement in the lower region of the pore as well, and the structure become a bit more slant, allowing the outer gate to be more open. Sorry, I didn't prepare the alignment of the structures on there, but we can talk at the cafe and I can show you on my laptop. I have the data. Perfect.
Hi, thank you very much for your talk. I have a question regarding KCA 3.1 channel.
Yes.
So there are structures solved, and in the very first paper from McKinn, they said that there is a open structure that they couldn't really very well solve. Let's say it was like around 4.5 angstrom resolution. Do you think adding PIP2 would help to stabilize this conformation?
So, wow, so if we mean as a structural biologist, I have heard from my colleague that if they really want to get the channel to bind to PIP2 and see the density, they have gone up to 50% PIP2 in the membrane composition. That's how Ji-sun got his case in Q1 with PIP2 bound with E1 in some of the work. So not E3, the E1, he has to really dump a lot of PIP2 in to get it to work. So it would probably work experimentally. Now, when people say that it's open, it is difficult to justify a channel that it's open. You see what I mean? Because open, is it open just that snapshot? Is it open because you built it to be open? Is it open stably in a simulation? You know, there are many ways to define whether the channel is open. Having a PIP2 there usually helps stabilize the open state if the channel is open by PIP2, but it needs to bind there, and it needs to bind there stably. And I think there is a challenge in the experiment there to get the PIP2 to bind stably. Some of the work that Rod has done also involve mutating residues near the PIP2 binding site to have more lysine and arginines so that PIP2 stick there better, and therefore leading to an opening of a channel. So there are several strategies you could play with the PIP2 binding site to really open, the pore. But yeah, it is. I'm not gonna judge whether a channel is open or closed in public. Yeah.
Thank you.
Thank you.
Okay, thank you very much.